Today we're talking about Chapter 5, Section 3, which is the regulation of the cell cycle. Before you continue with the video, please make sure that you have this Power Notes sheet. It's a concept map. We're talking about the cell cycle. You can see how we have different regulatory factors, both internal and external, and then most of the rest of it is focusing on cancer cells and what happens when that regulation goes wrong. So before you continue, make sure that you have something to take notes on. Now, the focus of this section is about cell cycle regulation and how it's necessary for the healthy growth of cells. What you see here in this picture, this is actually a fluorescence microscope image of some cancer cells that are dividing uncontrollably, and we'll talk about that a little bit later in this presentation about how that happens and what the body can do about it. So we're going to begin by talking about internal factors that regulate cell division. These are going to be proteins inside of the cell, and there's two categories of proteins that both work together to regulate the cell cycle. One is called cyclin, and the reason why it's called cyclin is because it is produced throughout the cell cycle, so it's constantly building up, and then it gets degraded as it's being used. So there's a cycle of buildup and then degradation, and that's where the name cyclin comes from. It's always present in those varying amounts. And then it works with another protein called CDK, which is a cyclin-dependent kinase. CDK is always present in a constant amount, and essentially it's activated by the presence of cyclin, and you can see that in the picture over here. The CDK in blue is always there. Cyclin is being produced, and as it builds up, more and more of it is available to bind to the CDK that's already there. When they are bound together, that makes the active form of the protein. And when that protein is active, it is allowing the cell to proceed from one stage of the cell cycle to the next stage. So essentially, there's different types of cyclin. Each one is responsible for a different stage of the cell cycle. And we're not really getting into the details of what all of those different types of cyclin are, so don't worry about that. Just understand that there are two categories of proteins inside of the cell that are responsible for regulating its division. Now, they do that at certain cell cycle checkpoints. The checkpoints are specific points in the cell cycle where the cell is not going to proceed unless everything is working correctly. Things like the DNA has been replicated correctly. Proteins are being made that needs to be made. If there's enough nutrients, if the cell has grown enough, all of those things are being checked for at the cell cycle checkpoints. And if everything checks out, if it's good to go, then the cell will proceed from one checkpoint and go into the next phase of the cell cycle. And again, the signal that it's using to go from one checkpoint to the next, it's using that cyclin and CDK combination. Now, there are some external factors as well that cells are responding to in cell division. In human cells, for example, they exhibit what we call anchorage dependence. And that's just a fancy way of saying that the cells must be attached to a surface. So even something like your blood cells, when those are formed, they're formed inside of your bones, in your bone marrow, which is attachment to a surface. And then after they have completed their cell cycle and they're just being used by the body, that is when they go into the bloodstream. The other thing you see here, density-dependent inhibition, that just means that the cells can tell when there are other cells surrounding them, and they stop dividing once they come into contact with those other cells. This diagram here shows a cut. So if these are your skin cells and you have a cut, there's a gap between cells. Well, these cells can sense that there's a gap, and that triggers them to undergo mitosis and cytokinesis so that they can fill it up like you see over here on the right. Now, once they fill this gap and they have cells surrounding them again, that is a signal for them to stop the cell cycle. They would enter G0 at this phase, and they would just do their normal function, but they would not keep dividing, or else that would result in a big mound of excess skin cells here. Now, when cells no longer can control their cell division the way that they're supposed to, that leads us to a disease that we know as cancer. Cancer is simply your own body cells that are undergoing uncontrolled cell division. And that can be really difficult to treat. Everybody probably knows somebody or has a close personal contact that has dealt with cancer before. This is a very pervasive disease these days because we're able to treat a lot of other diseases that historically would have been a problem in the past. Cancer is still notoriously difficult to treat because it is your own body cells, and you have to try to keep your healthy cells from being destroyed when you're trying to destroy the cells that are your own cells, but they are no longer responding to those cell cycle controls. So we do know that cancer is caused by exposure to certain substances that we call carcinogens. And a carcinogen is anything that is known to increase your likelihood of getting cancer. There's a few examples that you have here on the screen. Tobacco smoke has long been known to cause cancer. Even secondhand, if you're inhaling it, that's really bad. You want to minimize your exposure to tobacco smoke. It can cause lung cancer. Another one you may have heard of is asbestos, which is what you see here. This is actually a naturally occurring mineral, very long, thin fibers. And as long as it is inside of a rock or inside building materials, this is where it was used very commonly, it's fine. But once it becomes airborne, like if you are sanding something that has asbestos in it, that can become airborne. It can get into your lungs. That's very dangerous. That can also cause cancer. PCBs, or polychlorinated biphenyls, is what that stands for. You may have heard of these. This is an environmental toxin. It was used as a coolant. It's used in transformers. That's what this picture is. Those electrical transformers that you see on top of an electrical pole used to contain PCBs. Some of them out there still do. That's another cancer-causing chemical. And then obviously radiation. We know that radiation is bad, whether it's UV radiation from the sun, nuclear radiation, if we think of like a nuclear power plant disaster, that can also lead to cancer. So what can your body actually do about it? Well, first of all, there's a process called apoptosis. Apoptosis is programmed cell death. It is a normal feature of healthy organisms, and it's one of the major safeguards that we have against having these damaged cells take over our body. And the way it works is a cell that senses that it's no longer functioning correctly can actually produce an excess amount of these self-destructive enzymes and use those enzymes to digest itself. And the reason why apoptosis is an important process, first of all, it actually occurs as part of normal development. You can see in this picture here that all of our hands, as we were developing, started out with webbed fingers. But by the time you're born, that webbing has been destroyed. It has gone away, it has disintegrated, and that's because the cells have undergone apoptosis as a normal part of our development. So by the time you're born, you have 5 distinct fingers and no webbing in between them, most of the time. Now, if there is an error during development and apoptosis doesn't take place, then a baby could be born with webbed fingers, and that has happened before. That's an error in apoptosis. The other reason why apoptosis is important is obviously we were talking about cancer earlier. If a cell becomes cancerous and a cell is able to destroy itself, then that cancer is unlikely to spread throughout the body, and that is a very good thing for your body. Cancer cells, as they grow in your body, are very often going to form what we call tumors. A tumor is a clump of cells. It's not performing a normal function for your body. In fact, it can divert blood away from other important organs and tissues. These tumors fall into 2 categories. A benign tumor is generally considered non-cancerous because it's not going to spread throughout your body. It is a clump of cells that formed because they stopped responding to those normal cell cycle controls, but they don't spread throughout your body, and most of the time they're surgically removed with no problem. Malignant tumors, on the other hand, metastasize. Metastasis is when the clump of cells spreads throughout your body. So notice here, this picture is showing metastasis. This is a clump of cells forming a tumor. And then if some of these cells enter your bloodstream, just one cell here, it can flow throughout the rest of your body and end up in some other organ and start a new tumor in that new location. So again, we call that metastasis, or moving from one area of your body to another area of your body to form a new tumor. And by the time a tumor starts to spread like this, surgery is probably not the best option. This is when people undergo things like chemotherapy, which destroys cells using chemical drugs, or radiation treatment. If there are specific tumors that can be targeted through radiation, they can be destroyed that way as well. Now remember that cancer cell growth is not limited like normal cells. This is just a reminder here on the left of what normal mammalian cells do. They have to be anchored to a surface in order to reproduce. And once they reproduce to fill their space, they stop reproducing so that you don't have a massive clump of cells. If you remove a section, they reproduce to fill the section, but then they stop again. That's all normal. Cancer cells do not do that. Look at the picture over here for cancer cells. They have stopped responding to the contact of all of the other cells around them, and they continue to grow. They make this large mass of cells. This mass of cells would be a tumor in your body if it were growing in the body. And this is really why cancer cells are so dangerous, because they crowd out your healthy cells and they prevent your tissue and organs from working correctly. So when people die of cancer, what they normally are dying from is organ failure. As that cancer spreads throughout the body, the organs start failing. They're unable to function correctly. So cancer cells, our final thought here that I'm going to leave you with is that these are abnormal cells, both in terms of not responding to cell cycle controls, but also in terms of the genetic information inside of the cells. So if you look at this picture here, this shows what normal chromosomes are supposed to look like. This shows abnormal chromosomes that are found in a certain strain of cancer cells called HeLa cells. The HeLa story is actually a really interesting story that gets into questions about ethics and scientific research and medicine. There's a summary of that story here if you would like to read about it. It is fascinating, but it's a little beyond the scope of what we're going to get into today. But you can definitely see what the chromosomes are supposed to look like, 2 of each. And then these are the cancerous cells. We've got 3 of this chromosome. We've got 4 of this chromosome. We're missing chromosomes here. There's definitely a lot going on that shouldn't be in terms of the chromosomes. And so there's a big genetic component of cancer. In fact, there's a whole type of gene called oncogenes that are cancer-causing genes. If you have a mutation in a normal gene, that can turn it into a cancer gene, and it can cause your cells to reproduce way more than they should and form tumors and form cancer. So with that, we're going to end our discussion of Chapter 5, Section 3. Hopefully you found this to be helpful, and if you have any questions about what we've discussed here today, make sure you bring your note sheet to class so that we can answer those questions.